Hey guys, welcome to 11.4. We're talking about the final step in cellular communication, the response to a signal. And so here, I want you guys to remember that a response can be any number of different things. So it can be telling the body to release epinephrine, which we've spoken about, or to release a hormone, or to turn your genes on or to turn your genes off. So they would do this by initiating transcription or to stop transcription if you're going to turn off a gene. So there can be many different responses. And so there are four aspects to cellular response that we're going to talk about. One is amplification, which we've spoken about a little bit already, specificity and coordination, efficiency by scaffolding proteins, and then termination of the signal. So our first is amplification, which we've talked about a little bit already. So here, because a protein can stay activated long enough to work on substrate, more than one substrate, then it has the opportunity to increase the message or the size of the message. And so here, if we have, for each epinephrine that is being received, they can increase the message. It can act on 100 adenylyl cyclases. And so if you have 100 adenylyl cyclases, well then if they're working on, let's just say, each 100 cyclic AMPs, well then that message is going to increase to 10,000. So it's going to increase at each step, and so this is called amplification. Specificity and coordination. So epinephrine is going to act differently with on different types of cells. For example, if you have a heart cell, it's going to make the cell contract, which is going to allow less blood to go through at a moment, and then it's going to increase a rapid heartbeat or increase to a rapid heartbeat. Whereas liver cells, when they have received a chemical message from epinephrine, are going to break down glycogen. And so this specificity or difference in response is due to a different set of relay proteins within the cell. You can have the same some of the same proteins, but they're going to be doing different things. They're going to be in a different chain of events. And so that the response is going to be different. And then finally, efficiency by scaffolding proteins, or not quite finally, second to last, efficiency by scaffolding proteins. The cytosol, if you picture it, inside the cell is similar to the texture of jelly. And so if you have very small molecules, they'll be able to move quickly and easily through the cytosol. However, large molecules like proteins have a harder time. They're going to take more time to diffuse through the cytosol. And so, therefore, a pathway or a message could be slowed down by this texture, by this material. And so some pathways are more efficient because they have these scaffolding proteins, which are actually going to hold other proteins, 3 different protein kinases. And so it's going to allow the message to be passed on rapidly from one to the next. And so therefore, this is going to make the pathway more efficient. And now finally, for our last slide, termination. For a multicellular organism to remain capable of responding to a signal, it must last only a short time. So we saw when we looked at the cholera example, the GMP, the GMP was not able to be turned off, and so the homeostasis was lost because too much salt was allowed into the small intestines, which allowed too much water. And so therefore, we need to be able to turn off our pathways as quickly as they can be turned on. So this reversibility of a pathway, which we talked about with phosphatases and so on. So we need to be able to turn off a pathway just as quickly as we can turn on a pathway so that we can maintain our homeostasis. And that is it. I hope that you guys have a great day.